So for this week's lab, we had to make a concept map of plasma membrane and all the terms that were given to us. So here we begin with the plasma membrane, and here we see that we have a small little web for the vesicle. The vesicle simply made of membranes. And now moving on to the bigger part. Plasma membranes are all made of phospholipid bilayers. And that's kind of one of the big group categories in which I would divide this whole web as. So here we go directly into just more information about the phospholipid bilayers. First being that it has a hydrophilic head, and a hydrophobic tail, which are made of fatty acid chains. And that fatty acid chain is selectively permeable because it allows certain, depending on the fat solubility, it would allow it through a membrane. So that's kind of how I link that part. If we go down here, you see the structure of the phospholipid bilayer being two types structured. The fluid mosaic model and then the sandwich model are each two types of ways a phospholipid bilayer is formed or structured, I would say. From the fluid mosaic model, you would see that it is very fluid, and therefore it would needs stuff to make it fluid. Therefore, a steroid and sterol each create cholesterol, and cholesterol allows for fluidity within molecules and membranes. Going back to phospholipid bilayers, one major part of a phospholipid bilayer is that it moves ions through the membrane through its electrochemical gradient. This electrochemical gradient works with the membrane potential, which is the concentration gradient, and then there's two types of transportations in which the concentration gradient works through. The first being passive transport, and passive transport can be done through like membrane channels, which allows for membrane proteins to passively transport like ions and other molecules, as well as through osmosis. Osmosis is the passive transport of water, so those are two passive transport ways in which the concentration gradient works. We also have active transport, which just means that it needs energy in order to move things through its membrane, and then you would go into its ion channel, which in order to move ions through the ion channel, you would need some type of energy to push it through. And you have gated ion channels, which are similar to ion channels. The only thing is it has a specific chemical messenger that needs to interact with the site in order for it to allow for ions to go through. Coming back to the concentration gradient, the concentration gradient is very useful for moving ions in and out of the cell. If a cell has too many ions, which we would see down here, it would be known as a hypertonic solution within the cell. If it has too little ions, it would be hypotonic solution within the cell. And if it's just even compared to the outside of the cell, it would be isotonic, sorry. It would be isotonic. And from a hypertonic solution, here you would see the way you would get rid of a hypersonic solution would just be through simple diffusion, just more ions moving from the high concentration of the inside of the cell to the low concentration outside of the cell, which is something very normal through homeostasis in which everything would occur. Back to phospholipid bilayers, we're now going to see another major group in which this web was constructed around, and it would be membrane proteins. As you can see here, there are a bunch of different membrane proteins. First being detergent molecules assist make proteins more soluble, which allow for the membrane proteins to function. Here you go. Integral membrane proteins are permanent proteins that are attached to the membrane. You have passive membrane proteins, which are temporarily attached to the membrane, so they attach on and then attach off and replace, detach. Now you have a protein attached membrane associated with protein, being that it's proteins just that aren't attached directly to the membrane. They're either attached to a transmembrane, lipid-linked membrane, or a membrane associated. Now back to over here, we have also transport membranes, which move ions and molecules and macromolecules through the membrane protein, which work to function within channel-mediated passive transport. So that is another way of passive transport. Back over here, we have anchor membrane proteins, which basically just anchor the cell to the cell walls and of the matrix. And here we have enzyme membrane proteins, which are just enzymes embedded in the membrane, which allow for catalysis of reactions. Here we have monolayer associated membrane proteins, which are just proteins that interact with the inside, the hydrophobic part of the molecule only. And here we have lipid-linked membrane associated proteins, which are found at the surface of the membranes and attached through lipids. Here's the whole web once again. Hope that was what you were looking for. Thank you.